Hey everybody, how's it going? We're gonna jump in here to the second set of notes for the cells unit. They're called matter and energy. So we're gonna talk about how some material is dealt with in the cell, along with um, how it deals with energy. There's some questions for you here on the front slide. The relationship between matter and energy. Why does life require matter? Why does life require energy? And then how do interactions of cellular components allow the cell to process both matter and energy? So as we move through here, here's our to-do list. Okay, so this was on the last set of slides as well. Just kind of the major things the cell has to get done. We're looking at the second one here, processing energy, how to get and process nutrients, and also how to deal with gas exchange. Okay, so cells got to work to live. So the main ones here, got to make proteins, right? Because proteins control just about every cell function. Um, Energy is going to be important for daily activities at the cellular level, as well as at the organism level. And you have to be able to make more cells. You have to be able to grow. You have to be able to repair. Got to be able to go through mitosis, meiosis, and make new cells. So one of the organelles that deals a lot with matter is the lysosome. Okay. Now, this is an animal cell only thing. Plants do not have lysosomes. Okay. So it has several nicknames. The little stomach of the cell being one of them. I tend to refer to it as either the garbage disposal or the wood chipper because they kind of tear stuff up, or maybe the paper shredder. It's full of digestive enzymes, so that's what it's going to do. It's going to digest things. It's going to break them down. Okay, it's going to make polymers back into monomers. Okay, which can be helpful as well. It kind of works like a recycle center. It can digest worn out organelles. It can digest other molecules and recycle them, and so that they can be used again. Um, Once our immune system takes care of invaders for us, sometimes the lysosomes can dispose of the bodies and get rid of the remnants of bacteria or viruses, etc. So you can see kind of up here in the picture how the lysosome can merge with something and then go ahead and take it out if it needs to. A little bit more on lysosomes. They work best at a pH of around 5, okay, which is a little bit more acidic than neutral, right? So the organelle kind of creates its own custom pH, and I have some questions here, so not a bad idea to kind of pause the video and think about, why would these things be, or how would it get this things done. I mean, the lysosomes are breaking things down, so they have lots of digestive enzymes in there that are harsh, and a lot of those pHs tend to be a little bit lower, tend to be a little bit more acidic. It's also good to have them work at a different pH. If they were to somehow get out into the cytoplasm, then you might have a conformational change based on the pH, and they wouldn't digest the cell if you didn't want it to be digested. That could be a problem. If they escaped and they worked, they could be digesting lots of things that we still need. So it creates the custom pH. Well, how do you raise or lower the pH for anything? A lot of it has to do with hydrogens or hydroxides, right? So, adjusting how many hydrogens are in the area. So they have in the lysosomal membrane, pumps hydrogen ions from the cytosol into the lysosome. That's going to lower the pH. Enzymes are very sensitive to pH. We talked about this back in the first unit, right? Because the amino acids are charged. A lot of them have positive or negative charges, so that's going to dictate their structure and how they fold. And then if you change that, that can also adjust how they fold. Maybe they don't work as well. So enzymes are proteins, right? pH is going to affect structure. Why evolve digestive enzymes which function at a pH different from the cytosol? Well, I kind of went over that already, so that if you do have some sort of leak, then they're not going to work very well, and that will save itself from being digested itself. Okay, so there you go. So when things go bad, if they're supposed to get rid of things for us and keep things clean, right, kind of like a garbage disposal and get rid of stuff, then if the lysosome doesn't work well, then it's not going to get rid of stuff, and stuff's going to build up, right? That means you're going to have a lot of material in there that's undigested that should be gone but isn't. And whenever you have a buildup of anything really in the body, it can really cause a problem. It can disrupt the function of other organelles or other organs or whatever the case may be. So lots of lysosome diseases, unfortunately, about 40 or more known. One example is Tay-Sachs. That's probably one of the more well-known ones. And it's buildup of undigested fat in the brain cells. You can see this little boy has Tay-Sachs. They generally don't have really long lifespans, and they generally have some problems with their neural development. So you can definitely have some issues if your lysosomes have some sort of mutation or aren't working properly. Lysosomes can be good. Killing cells can be a good thing. Sometimes, they have to go through apoptosis, which you might remember from regular bio. Sometimes it's just for development. So like tadpoles into frogs, the tail, it doesn't just fall off at the base. It kind of works backwards from the tip, and then cells tend to start going through apoptosis. And then the cells are destructed all the way back until the tail is gone. And so that can happen. There's a few cases like that in the natural world where apoptosis is responsible for that. That's one way. Also in humans, if you look right here at this picture, here's an eye, here's an arm, and this is the hand. And you can look kind of closely in there and you can see where the fingers would be, at least some of the fingers in there as they're forming. And so look how much webbing is between the fingers, and we all have a little bit of webbing down here. Some people have more than others, but all the webbing that was in here gets chewed out by apoptosis, and it gets worked out of there in between all of our fingers, forming the digits. So it happens with our hands and feet that we start off with a lot more webbing in between there than what we end up with, and apoptosis takes care of that. Okay, so a lot of times we have cells that also need to die. We have programmed cell death is what we call it, or apoptosis. And basically, if cells are growing uncontrollably, then we need to stop that. Okay, we don't want cells to divide and divide and divide and divide. When they do that, then they tend to make tumors. They tend to make piles. Tumors are just piles of cells that have divided too many times, right? And this can ultimately end up being cancerous. Not all the time, but it can be. And so sometimes you have to kill one cell for the good of the many. So you kind of have to triage on the battlefield, right? You have to make sure if the body or if the cell detects that there's a problem in the cell cycle, then it will kick it into apoptosis and go ahead and digest that cell so that it doesn't become a bigger problem down the road. And to do all of their stuff, cells need power, right? So we're going to talk a little bit about energy now. So you take in food, you digest it, you take in oxygen, you make some ATP, and you remove waste, right? That's a big process called cellular respiration, right? So why do cells need this energy? Well, you got to have energy to do work, right? That's kind of the definition. And so where is that energy stored? And if you guys remember from the first unit, you guys should know that it is stored in the bonds, okay? And why ATP? Why this particular molecule? Because it's a pretty handy molecule, actually. If you look at it, it's very similar to one of the organic molecules you guys studied in the first unit. So that would be you have your carbs, you have your lipids, you have your nucleics, and you have your proteins. Which one of those four molecules looks like this, where you have a sugar like a ribose, and you have a base like an adenine, and then you have some phosphates over here. So hopefully that should start ringing bells that's like nucleic acid. Okay, so adenosine triphosphate, ATP, adenosine triphosphate, 1, 2, 3 phosphates. Okay, so it's very similar to a nucleotide, just a little bit different. And that's our energy molecule. And what happens is, we're going to get into this, but the energy is stored in those bonds between those phosphates. And that's how it becomes kind of like a rechargeable battery. So cells have to do this converting, right? They have to convert or process the incoming energy into forms that it can use. So we're gonna do some basics on respiration and photosynthesis. This will become its whole other unit later this semester. So our last unit this semester will be on this stuff. So we're just going to kind of stay on the surface as we talk about cellular energy, but we do have to talk about this here as well. Make it a little bit easier down the road. So mitochondria going from glucose to ATP. Okay, that's pretty much the job. Chloroplasts are using sunlight, and they're going to generate some ATP and carbohydrates. Okay, so two different organelles there that are going to go about things different ways, but ultimately are trying to generate some energy. Okay, so important to see the similarities between these two. So mitochondria and chloroplast. You might be tempted to say that one's in an animal cell and one's in a plant cell. That would not be entirely accurate. Okay, mitochondria are in animal cells and chloroplasts are in plant cells. Okay, but do not forget that plants have mitochondria too. Okay, plants have both, so make sure you don't forget that, whether it's this unit or the next one. Okay, make sure you know plants have mitochondria too, and I'll be reminding you of that as we go, and we'll talk a little bit about why down the road. So they both transform energy, they both generate ATP, they both have two membranes. They are both semi-autonomous organelles. And if you have autonomy, what do you have? Well, you're kind of your own boss. Okay, so some of your parents, if you ask them, do you have autonomy in your job? That means, do they get to make decisions? Do they get to kind of, do what they want, or, are they regimented on what they do? Meaning like, other organelles are more controlled by the nucleus, where these guys kind of get to do their own thing because they can move, they can change shape, they can divide. They were their own free living organisms, right? And so they have their own DNA, they have their own proteins, they can do a lot of their own stuff, despite the nucleus, um, not necessarily needing to be there to control them. So, like I mentioned, internal ribosomes, DNA, and enzymes. So mitochondria functions in cellular respiration, again, to generate ATP. And the gist, the basics is you break down sugars and fats and other fuels. You break down your food, right, in the presence of oxygen. So why we have to take in oxygen. We need it for the reaction. We'll get to the specifics of why oxygen down the road. But you break down larger organisms into smaller ones, which is the catabolic side of things, if you remember from our reactions in the first unit. And then because we're doing this in the presence of oxygen, this is an aerobic process, right? Aerobic respiration, aerobic reactions. So if we look at the structure of a mitochondria, we can pick out a few things here. Let me move my head over here. And obviously it has two membranes, and it has, the big oval one going around the outside, which is kind of typical of any, organelle, where you just have this one going around the outside here. And so that's a regular, outer membrane there. But then you have this other kind of really convoluted one in here that's all curvy and wavy. And I know my line's not exactly what their lines, but you get the idea. And that's called the cristae. Okay, the inner membrane is called the cristae. And that's where a lot of reactions take place. And it's all convoluted and curvy and all of that because it provides surface area so that more reactions can take place. Specifically, the electron transport chain takes place there, if you remember that term and process from regular bio. And that allows for a lot of ETCs to be happening all at the same time, so we can make more ATP. Okay, so rid of my pen. And then the mitochondrial matrix, you can tell from the diagram, that's all the space in between where you can see all the ribosomes in there that are making proteins. But yeah, the matrix is just all the space inside the cristae. Okay. There's your answer for why two membranes, which I already told you. So just another picture kind of side by side on the cartoon version of the mitochondria versus the real one. And you can pause the video and look at the parts if you want to. No details to know here. This is just a reminder of the ETC that does happen in this membrane of the mitochondria. You will need to know this in great detail in the next unit. This one, I won't ask you the details on it, okay? So let's just look real quick. So inner mitochondrial membrane right here, okay? So this whole membrane right through this area, okay? That's really one of those lines of the cristae, okay? And so we have the matrix on the inside, right? Here's the matrix, and then here is the inner membrane space all up here. And so what's happening is we're pumping a bunch of hydrogens out, and then it's that those hydrogens are going to come back through, and they're going to come back through this way, and they're going to be used to power this guy, ATP synthase. Okay, that's the right. It's an ACE, so it's an enzyme. It's synthesizing ATP, and that's how we make it. Okay, so I'm not going to go into any more details than that. We'll talk about all the other things on this slide down the road. Here is your equation though for respiration, right? You need sugar, you need food, you need the fuel, plus the oxygen, and then you get CO2, water, and energy. Okay, so inputs and outputs, you might have to deal with those for now, but no details. And there's the actual equation written out. Sugar, oxygen, CO2, water, ATP. And you have to balance that equation as well, which is why you see the 6's in front of 3 of those. So mitochondria can divide on their own. We mentioned that. So who else divides like that? Bacteria did that. They do that through binary fission, right? Where one cell copies the DNA and turns into 2. Well, this is how mitochondria do it. So this is just a reminder that, of the endosymbiont theory, and through evolution, these guys were once their own free living things, and then they were engulfed, but they retained a lot of these own individual properties for themselves. So few more details on the mitochondria. There could be one, there could be hundreds, could be thousands. It really depends. I think I've mentioned before that, cross-country runners in your high school are going to have more because they put more of a demand on their body for aerobic respiration. And so it doesn't mean they're going to have more cells than you. They can have more mitochondria per cell because the demand is there. Okay. So it can really be correlated to that. More activity, more energy needed, more mitochondria. Okay. So you probably could think of this on your own, but the most active cells, right? Muscle cells are going to be really active. Nerve cells are going to be really active. So they're going to have lots of mitochondria. Mitochondria are everywhere. This is just that reminder that you see them in both. Okay. Here's the animal cell over here, and here's our big mitochondria. There's a few right there, but they're also over here. They have mitochondria in the plant cell as well. And so, the way I kind of want you to think about this is, there are no chloroplasts in an animal cell, so we have the mitochondria for respiration. But if we think about the chloroplast and we think about photosynthesis, okay, what does photosynthesis actually produce? Okay, and you may not quite remember. I'm about to tell you in some slides here in a second. But what do the chloroplasts actually produce? And the answer is sugar. Okay, so it converts sunlight into carbohydrates. And so if they didn't have mitochondria, the only thing the plant would be able to do is build sugar. Well, if you just build sugar and build sugar and build sugar, do you get any energy out of that? No, because you don't have anything to break it down. The mitochondria is there to run cellular respiration inside plants so that they can break down the sugar and then get the ATP out of it. So they have to run both processes. Plants run photosynthesis and they run cellular respiration. Okay, really important. Some people just, don't quite get that they do both. Okay, so make sure you're getting that. There's some circles for you. So a little bit more about chloroplasts. Chloroplasts are only one part or one example of a plastid. There are other plasts out there. There are amyloplasts, which store starch in roots and tubers, kind of like potatoes. You have chromoplasts that store pigments or colors. You have the chloroplast, obviously, it's going to store chlorophyll. That's the molecule inside the chloroplast where a lot of reactions are going to take place. And it also is kind of that pigment in there, gives them their green color. And we'll get to some of these parts over here in a different picture. So here's some structure for you. So if you look at some of the parts, you have the stroma. Better get a pen. You have the stroma, which is actually in this picture, it's all this orange space, okay? All the space in here that's orange is the stroma, okay? The thylakoids or the thylakoid membranes, that's all these stacks, so all these discs in here, okay, which sometimes I like to call pancakes, okay? So the thylakoids are the pancakes there, and the grana, okay, grana is the stacks of thylakoids. So grana is one whole stack. And you might see grana with an A, or you might see granum with a UM. Granum is one stack, grana is plural. Okay, so that's why you might see a couple different spellings of what you think might be the same word, but it's just a singular plural thing. Okay, grana is plural. So again, why so many stacks of thylakoids in there? Because they have a lot of membranes, and this is what's going to increase surface area for all the reactions that are going to take place. Okay, surface area on membranes, super important for reactions in a cell. Here is a little refresher of photosynthesis for you. You do not need to know any details for this unit. You will need to know tons of details for next unit. Okay, so right now I'm going to try to stay big picture here, and I might even just stick with this top one up here. If you look, there are two real parts to photosynthesis. There are the light reactions, and there is the Calvin cycle. And the light reactions, we are taking in light and water, and we are doing a couple things. We're converting light energy into chemical energy. Light to chemical, light to chemical. So light energy to chemical energy is what's going on in the light reactions. And we are taking in water to help with that. And we are giving off, I say we, plants are giving off O2. Okay, and that O2 will go out for us. Okay, so then the chemical energy that we convert, remember sunlight to chemical energy right here, then we use this energy to power, wow, that's a bad circle, but we are going to power the Calvin cycle, which is the sugar building reactions. Okay, so convert the energy from light to chemical in order to build sugars. That's the goal. And we use CO2, or plants use CO2 to build the sugar. Okay, so I'm not going to mess with the bottom picture at all right now. We will get to that down the road, as you don't need to know any details for that at the moment. Here's your equations, and you should remember that they are mirror images of the respiration equation. They are really just flip-flop, where the products of one are the reactants of the other. So a little bit more about chloroplasts. So obviously it's involved in photosynthesis. These are anabolic reactions. They are building, right? We're building sugars, we're building molecules. So you're taking CO2 and you're building CO2 into a sugar, okay? Again, semi-autonomous, can move, change shape, divide, reproduce. It can do things all on its own, right? Just like bacteria, just more evidence about it being its own thing. And real quick, some more properties about them. So why are they green? Just kind of a reminder about how sunlight works and why these things are green. So if you have the sun up here and all of the colors are coming in, right? All of the colors of the rainbow come in, and we only see green in the thylakoid. That means all the colors were absorbed except, notice the green arrow coming out. I probably should have used a green pen. The green arrows are coming out because they are getting, they either pass through or they get reflected. And that's why we see green into our eyes, right? If someone in your house has a red shirt on, that red shirt is absorbing every color in the rainbow except for red. The dye in the shirt won't absorb red, so it gets reflected back into our eye, and that's what we interpret as red. Okay. So here is just a kind of a refresher of a plant cell, and most of the organelles are the same. There are a few differences. Grab this. So, if you look at this, nuclear envelope, nucleolus, chromatin, nucleus, all the same that we've talked about. Rough ER, smooth ER, ribosomes. Central vacuole is really just reserved for the plant, really, because it's this big clear guy, and it's really just there to store water. And so that's an important one for plants. We don't talk about tonoplast a lot. I wouldn't worry about that one. And we haven't gotten to the cytoskeleton yet, but we will. And these are all the same. Microfilaments, intermediates, microtubules, those are all the same. Chloroplasts, obviously, only for plants. Plasmodesmata, we haven't gotten to yet. Those are just pores. Then you have the cell wall, not in an animal cell, right? Okay, only in a plant cell. You have the plasma membrane, the peroxisomes we've mentioned, mitochondria, Golgi. So lots and lots of similarities between plants and animals, just a few different ones, okay? So here's an important thing down here, not in plant cells. So things that were in animal cells, lysosomes, centrioles, and flagella, okay? So a couple differences there between things that are in animal cells and not in plant cells. So mitochondria and chloroplasts are different from the other organelles. Move that over here. And they're not part of the endomembrane system like a lot of the other organelles. They can grow and reproduce. We said they're semi-autonomous. Their proteins are primarily from the free ribosomes in the cytosol. They also have a few of their own. They have their own circular chromosome, just like bacteria do, right? One singular circular chromosome, and they have that. And so, again, more evidence for them being their own thing once upon a time. And this is just kind of a reminder of the endosymbiont theory, where they were once free-living bacteria. They were engulfed by an ancestral eukaryote. And then they've lived on in a partnership, right? Whereas it's an evolutionary advantage to both. One supplies the energy and the other supplies the raw materials and the protection. So the cells provide the raw materials and the protection, and the organelles, the mitochondria and chloroplasts, give that cell energy. This is one of the researchers that figured this out, Lynn Margulis, University of Massachusetts, Amherst. So this is just a picture showing you kind of how it went. So here's your ancestral prokaryote, and you can see like here's the folding in of the plasma membrane, and then one of them turned into an ER, and you get the nuclear envelope and the nucleus, and so it's progressing along the way. Years, this is not quick, right? Lots of years have gone by, thousands and millions of years have gone by. So here we engulf an aerobic heterotrophic prokaryote. Okay, so this is the mitochondria, right? So that gets engulfed first, and then it carries on, and this is how the lineage goes for animal cells, right? Heterotrophic eukaryotes, okay? And then up here, this one also has the mitochondria, but then it also engulfed photosynthetic prokaryotes, and it engulfed chloroplasts, and now we basically have the photosynthetic eukaryote lineage up here, which are going to be your plants. Okay, so hopefully that makes sense for you. Again, the equations are just mirror images of each other. You can pause the video and check that out. You should have seen this in at least one, or not, if not two or three other classes before now. Pause the video and look at it if you need to, but very much mirror images of each other. And it really does create this circle of life, right? So the sun comes in, the plants do photosynthesis, right? Without this interaction, without photosynthesis, really nothing on the planet is going to work. The planet is a system, okay? And so plants convert the sun's energy into chemical energy, and they get glucose and sugar for themselves, and then they give us oxygen, right? If that didn't happen, the planet wouldn't be oxygenated, and animals would not be able to get any energy for themselves, because we need oxygen to do cellular respiration. It doesn't happen without it. So plants and animals do cellular respiration, and they get some energy for themselves, right? And in that process, we give back CO2, which then the plants have to have in order to do photosynthesis. Without CO2, they can't do photosynthesis. So it's, they're very reliant on each other. It's the only way this works, and it's the only way the planet works. This is not just, a simple reaction between, humans and your dog, okay? The plants and the grass and the trees that you see, like, this is running the entire planet, okay? This whole interaction is running the entire planet. So you have to have all of this going in order for the planet to work. Few other things that we're going to throw in here. Plant cells, I mentioned the central vacuole, right? So that's going to be for water storage. There's also what we call a contractile vacuole in some organisms. This one down here in the center happens to be a paramecium, which is a type of protist. Um, and you can see it kind of looks like your hand, and the center part is where it brings in water, and it's kind of the storage stuff. But these things live in water. So if they live in water, water will constantly want to come in, right, through osmosis. So in order to not be completely overwhelmed by water, they have to contract, and it contracts like this, and it's basically pumping water back out so that it doesn't get completely flooded with water. So they have contractile vacuoles to kind of manage how much water is inside the cell or the organism. In this case, which is the same thing, single-celled organism. Okay. And then up here, you also see some food vacuoles. Okay, you have a lot of vesicles that you could call food vacuoles, and how things pinch in and they bring food or molecules in, which then usually get hit with a lysosome and start digesting whatever gets brought in. So here again, food vacuoles, contractile vacuoles, central vacuoles. Um, Phagocytosis is cell eating, right? So that's bigger things, bigger molecules that get brought in, and they fuse with the lysosome, so the lysosome can digest them. Contractile vacuoles are in freshwater protists, and they pump out the excess water, like I mentioned. And then central vacuoles, plants have these in a lot of their cells, so they can store water. So they can do photosynthesis. Even when it's not raining, they'll have enough water in their cells to do photosynthesis. We also have some vacuoles for storage, and it can store a lot of different things. It can store proteins, it can store metabolic byproducts, pigments, defensive compounds, uh, tons of stuff, right? Literally tons of stuff. And these have selective membranes, which is going to control what goes in or out. And so lots of different functions for vacuoles. Putting it all together, animal cells, plant cells, you can run through the organelles on these pictures if you want. Remember that mitochondria is in both. Okay, that's an important one. But you can kind of go through organelles if you want to on that guy. And then here are some review questions for you when you get ready to go through that. So hope that was helpful. And we'll be going into some labs next week for the in-person kids. For you guys, I'm not entirely sure how this is going to go yet. I'm still kind of working through some things on how to make labs work for you guys, because while we've done computer labs up to this point, we're getting ready to get into some more real biology labs. So I'm working on some things for you guys. It is not going to be the same. You guys are not going to be able to do some of the hands-on stuff that the kids in class are doing. So you're probably going to get data, or you're going to get worksheets that mimic it, maybe some videos, but it's just not going to be the same for you guys. And I'm not sure how else to deal with that, but we'll do the best we can. Okay. All right. Take care.